Welcome, everyone. Thank you for joining us for today's webinar, Developing Precision Combination Therapies to Overcome Evolution of Therapeutic Resistance, presented by Dr. Khyati Shah, postdoctoral research scholar at University of California, San Francisco. My name is Xavier Gutierrez, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Shah. I will now turn the presentation over to her.
Thank you, Xavier, and thank you, LabRoots, for giving me this opportunity for presenting my work. So since this is our drug discovery symposium, I'm going to talk about how we can develop the precision combination therapy and how that can help overcome the therapeutic resistance. And to get started, I will just give the outline of my talk. So first, I will introduce why the drug resistance is a prevalent problem in drug discovery. What are the strategies by which we can overcome the therapeutic resistance, and overview of drug combination to enhance drug discovery. And as an example of designing the rational drug combination, I will give an example how therapeutic resistance to EGFR TKI can be targeted to utilize this combination and develop further strategy. So to do that, I will talk about my model systems that we developed to model the acquired resistance and how we can use the high content microscopy to discover the combination therapy. I will also talk about our results that we identified Aurora kinase A as a synthetic lethal target to overcome resistance. And at the end, I will give my conclusion. So let's get started with this. So just to give you the current landscape of drug discovery, as you can see from year 1993 to 2018, there is increase in amount of new molecule entity or biological entity that has been filed to FDA for drug approval. And 2018 was the highest, where we saw 14 new drug application and 17 biological applications for drug approval, and that's a huge. If you overall look at the trend of drug discovery, especially in case of oncology saw highest amount of drug approval or the drugs that enter into the clinical trial. However, there are so many molecules that enter the clinical trial, but not all of them succeed. And the reason being there is a high attrition rate, meaning that the drug that enter the clinical trial doesn't get approved. And that attrition rate is highest in oncology. There are various reasons behind this high attrition rate. A, there is a limitation of efficacy. B, there could be toxicity that is not discovered in preclinical target, lack of efficacy, lack of biomarker, lack of knowing the right population to target the drug, and hence for various reasons the drug discovery has seen the small amount of success in terms of getting drug approved. So what can we do about it? We are getting better at this. So now in the case of a post-genomic era, we can classify the tumor based on its oncogenic genetic driver. And in case of a non-small cell lung cancer you can see that based on the genomic driver, tumor can be classified, and there are major drivers such as KRAS, EGFR, ALK activating mutations that happen that drives the oncogenesis of non-small cell lung cancer. And I will focus for this purpose only on EGFR. So EGFR is known as an epithelial growth factor receptor, and it drives the cell proliferation in non-small cell lung cancer. So there are various inhibitors that are designed to prevent this hyperactivity of EGFR. And some example of these EGFR TKIs are erlotinib, afatinib, and most recently, osimertinib. So just to give you an example of osimertinib progression in the clinic, osimertinib was first synthesized in 2009, and in 2013, it entered clinical trials. This is a very unconventional way of getting into the drug trial in a very short amount of time, but this drug was highly efficacious. And when it entered into the clinical trial in the phase one itself, there was a demonstration of efficacy, and US FDA approved this in the subset of patients with non-small cell lung cancer who have resistance mutations in T790M. And hence, the drug got fast-tracked approval in 2014. And then in 2016, it was approved as a first-line therapy for patients with EGFR mutations. And overall, if you see, the total cost of drug development for osimertinib was about $2.6 billion. And currently, the annual sale of this drug is $1.7 billion. And it's expected to rise by $10 billion in 2025. So there is a huge success benefit available to the patient who harbor these EGFR mutations in them. However, if you look at the waterfall curves of these patients, so what are waterfall curves? Waterfall curves over here indicates the response of the patient to that particular therapy, and each bar over here is an individual patient. So what you can see that the majority of the patient have either unconfirmed partial response or they have a standard disease that they progress on. Very few patients eventually receive complete response. So A, this response is not durable, and B, there is never a complete response, meaning that this therapy, although is very successful, has a major limitation in terms of acquired resistance. Does this happen in the other cancer? And the answer is yes. What you can see here in the picture is the patient with the melanoma. When they were put in BRAF, they harbor BRAF V600E mutations. And when you see that when the patient before the therapy in A was on, at the time of diagnosis, when the patient was put on the melanoma vemurafenib therapy in 15 weeks, the patient saw like regression of the tumor and almost normal looking morphology over here. But the patient on therapy progressed within 7 weeks in such a short amount of time with even more aggressive tumors. So it's clear that the resistance is a major bottleneck of any success of the therapy. Recently, there is a lot of excitement with the approval of immunotherapy, and the resistance is prevalent in the case of immunotherapy also. So here is the example of anti-CTLA-4. Majority of the patient, despite having the CTLA-4 marker, they do not respond to the therapy well. What you can see on the left side of the panel. And in the case of the right side of the panel, the patient, there are subset of the patient who respond, but they almost never receive the complete benefit or complete cure out of this therapy. PD-L1 also has a similar trend, meaning that the majority of the patient who respond, A, do not achieve the long-lasting benefit, and B, there is emergence of resistance in for the therapy. So just to give you the flavor that most of the patients with advanced cancer die because of the cancer exhibits or develops a resistance to the therapy that they are taking. And hence there are paradigm shifting efforts are needed to understand and combat the resistance and to discover the mechanisms that is underlying the resistance. So therapeutic resistance can be mainly classified as primary resistance or acquired resistance. In case of a primary resistance, these are the patients who were on the left side of the waterfall curve, meaning that they do not show any benefit from initial drug therapy, despite having the positive markers for it. And these patients could have like a tumor intrinsic factor and activation of alternative pathways downstream, or there could be a patient-specific factor, meaning that the patient have either drug-drug interactions or are metabolizing the drug very fast, and hence the drug is not available for the activity. However, majority of the patients do respond initially, but they have an incomplete response. And the factors that causes this incomplete response and eventually progression consist of makes it to the acquired resistance. And acquired resistance could displayed in a 4 flavor. There could be the target modifications. So the drug that we are trying to target could modify and exhibit like a secondary site mutations, causing the inactivation of drug binding pocket. There could be a bypass signaling, meaning that there could be a compensatory loop that can be activated, and hence this pathway is hyperactive and is the drug has no efficacy in that case. There could be an example of histological transformation, meaning that the patient tumor completely transform into a new phenotype, or there could be the other mechanisms such as increase in the growth factor productions. So now that I have given you the flavors of what kind of therapeutic resistance mechanisms are seen in the clinic, there has to be a strategy to overcome this. So first I will focus on the primary resistance. So the patient, when they display the tumor intrinsic factors, there could be the tumor intrinsic factor can also be targeted by dual target blockade. And mostly this is achieved by drug combination. If there are patient-specific factors, then we need to modify the dose or modify the schedule, how the patient is receiving the drug. In case of acquired resistance, whether it is target modifications, patients can try the next generation EGFR inhibitor, inhibitors, such as in case of EGFR. If the patient progresses on orlatinib, they take osimertinib, which is efficacious in inhibiting this target modification of T790M. So clearly the drug combination works in such patients. If there is a bypass signaling, then again the dual target blockade is needed for the pathway that is alternatively activated upon therapy. And to target that also we use drug combination. If there is a histological transformation, we need to identify what change occurred in this tumor and how this tumor has developed the synthetic lethality in them. And that can also be targeted with the drug combination. So clearly this suggests that for the targeted therapy, the drug combination works the best in order to inhibit the resistance. So the question is, does this happen only in resistance happens only in targeted therapy, or it happens in immuno-oncology? And the answer is it can happen to immuno-oncotherapeutics as well. If you look at the common mechanism, resistance mechanisms, in case of immuno-oncotherapeutics, it's mainly either primary resistance, meaning that the patient samples have poor infiltrations of the immune component. There could be acquired resistance caused by loss of the antigen presentation on them. There could be the resistance because of the alterations of chemokines. There could be overexpression of VEGF that could lead to altered T-cell trafficking into the tumor formation. There are various factors also. There could be like a modulation of co-stimulatory pathway that upon PD-L1 inhibition that could lead to the primary resistance. And there are various mutations in JAK1, JAK2 pathway or upregulation of PD-L1 or upregulation of other immune checkpoint markers that causes the acquired resistance to the immunotherapy. And hence, to do that, to counteract this therapeutic resistance to immuno-oncotherapeutics, people have tried various combinations therapy to target the various modalities of the immune component, such as myeloid component can be targeted with the IDO inhibitor or anti-CD40 inhibitor. The T effector cells can be modulated by the combination of either PD-L1, CTLA4 inhibition, or anti-LAG3 antibodies. The Tregs can also be modified, and the tumor intrinsic factors can be modified with the help of combination therapy, targeted therapy, or chemotherapy. So clearly, the combination therapy exerts a lot of benefit in order to overcome the acquired resistance to targeted therapy and to immunotherapy. So is this resistance mechanism and the combination is historically very well known. And what you can see over here that in the history of the drug development, initially people have identified the methods by which you can find out whether the drug has a synergistic effect or whether the drug has the additive effect. And to make, in terms of drug discovery, when the BCR-ABL was discovered, there were the resistance mechanisms that were also identified with it. And as you can see in the pathway of drug discovery at various stages, the acquired resistance as well as the complementary targeted drug combination has been discovered. And the advantages of it, what you can see over here. So what I have shown you earlier is that the patients do not respond to the single agent immunotherapy very well. But in this case, when you combine the anti-CTLA4 and anti-PD-L1 inhibitor, you can see that these patients not only derive like a better benefit of it, but majority of the patient respond, showing that most of the patient fall on the right side of the curve rather than the left side of the curve. So the combination therapy has a lot of advantage to it. In case of a targeted therapy, these are the melanoma patient that I showed you earlier in the picture that were progressing. But now when they take a combination of BRAF-V inhibitors such as pembrolizumab and MEP kinase inhibitor, which is the alternate pathway that is activated, almost all the patients do respond initially to the combination very well. And hence, it's been shown in the field that the chemotherapy and targeted therapy or both the immunotherapy by itself do not have a strong capability to induce complete response. But the combination shown over here of ipilimumab and PD-1 blockade can enhance the response. But now that we know what would be the other resistance mechanisms, so we can design like a suitable triple combination along with the targeted therapy or with the chemotherapy to drive, to enhance the survival benefit of this drug. And to do that, there are currently about 2,250 active clinical trials going on along with the different immunotherapy on x-axis, and on y-axis you can see the different modalities that can be combined with this to maximize the drug response. So why we do this? The reason, because we can enhance the efficacy of the drug, and hence it can have a patient survival benefit, as well as it can increase the profitability of the pharmaceutical industry. And hence there is a lot of investment by pharmaceutical companies to discover this combination and study the resistance mechanisms. So now that I have given you the background of why drug resistance is a bigger problem and why combination can potentially overcome this, I would give go into the more specific example with the case of EGFR TKI. So over here, you can see the patient at the time of diagnosis, at the baseline, have a tumor burden in it. When the patient is treated with the AZD9291, which is the osimertinib, which is the EGFR TKI, patient shows maximal response, and there is only a residual disease, a very tiny tumor left in the middle panel. But when the patient was kept on the osimertinib, within 10 to 12 months, they developed the acquired resistance. And now that in the presence of drug, their tumor were actively proliferating. And these resistance mechanisms are very well known in case of a EGFR TKI. So what you can see over here, that the resistance mechanisms to first generation EGFR inhibitors are MET amplification, HER2 amplification, XL upregulation. There could be mutations in PI3 kinase and BRAF pathway. There could be EMT, epithelial to mesenchymal transformation, as well as small cell looking morphology. These patients receive third generation EGFR inhibitor, such as osimertinib, the example that I have shown. But the patient also relapsed to that with either the known genetic mechanisms or with unknown genetic mechanisms. And these patients have no effective salvage therapies available to them. Meaning that if you are a patient on EGFR TKI and if you progress upon the third generation EGFR inhibitor, osimertinib, there is no option for you other than chemo. And these tumors are immunorefractory, meaning that there is no other option. So how the resistance is monitored in the clinic? It's mainly monitored with the liquid biopsy. So the patient who initially, when diagnosed with the EGFR activating mutations, they take the blood sample and they do the liquid biopsy to see what is the prevalence of EGFR mutations. Once the patient progresses on the first generation EGFR inhibitor, as you can see that there is a steady increase in the EGFR T790M, which is the resistance clone. And at the intermediate step, this resistance mechanism is prevalent, and these patients get put on to the third generation EGFR inhibitor. And once they progress on third generation EGFR inhibitor, you can see their mutations profile. The P53 mutation load goes up. There is emergence of the secondary mutations in EGFR C797S also getting up. So this tumor is constantly evolving upon the progression of the therapy. And the tumor evolution is a driving force of clonal diversification. These readouts that we measure in the liquid biopsy comes from the different clones that are present in the tumors. And how these clone diversification initiates, it's not known. And what are the strategy to prevent the evolution is also unknown. It's being thought that since we see the various varieties of the clones which evolve over a period of time, we can target multiple clones. But these resistance mechanism often happens in the same tumor, and you cannot design polytherapy to target like 5 different clones. It's not favorable in terms of patient A, in terms of cost, and B, in terms of toxicity. So what are the ways to inhibit this clonal diversification and tumor evolution? We wanted to study this, and to study this, we developed the clinical equivalence model. To study this EGFR TKI resistance. So initially, when the tumor is sensitive, you give them the EGFR TKI, and in vitro, most of the cells die. So you can see the steep decline from day 0 to day 3 upon the EGFR therapy, what happens in the patient. But eventually, upon maximal response, there are some drug-tolerant cells that are left in the patient tumor, and this gives rise to a residual disease. And ultimately, these residual disease eventually progress and display in terms of acquired resistance. So we can clearly model this in vitro. Here you can see the cells enters the residual or drug tolerant phase, and at the end of between 3 to 9 days, and after when you keep them on drug, after 6 weeks, these cells start proliferating again in presence of drug. And to study this, we took like 4 drug-sensitive cell lines treated with the osimertinib to develop the acquired resistant cell lines. And here I'm just giving an example of two models. So the PC9 and 1975 are the parental cells indicated here in green, and they are sensitive to the 2 different third generation EGFR inhibitor. But when you take the acquired resistant cells, they can tolerate the high amount of drug and they grow fine in presence of drug. So since we saw the benefit of studying the drug combination, I'm just giving a practical considerations that we can take into account to identify what are the combinations that can and how we can design the combinations. So the basic methodology to study the synergistic action is to do the drug response matrix, where you take the 2 drug and you ask the combined inhibition, and then you calculate the combination index using following methods in a high throughput screening way. Or you can have a refined genetic or a pharmacological perturb systems where you study the drug-drug interactions or drug-gene interactions or gene-gene interactions using either chemical library, PROTAC inhibitors, or RNAi library or CRISPR. But the point of these therapy is to identify the synthetic lethality or a dependency that has been altered in the cells. And once you identify these combinations or a genetic dependency or non-genetic dependency for that matter, we need to validate that. And to validate that, we primarily use either the cells ex vivo in cell culture, or we use PDX or xenograft or organoid models to verify the synergistic effect of the combination. So there are different types of combinations. First is a synergistic or additive combination. How that have an effect of drug on drug resistance is either they can prevent the pathway reactivation, or it can prevent the downstream pathway inhibition, or it can cause the inhibition of parallel or compensatory pathways. With the help of RNAi or CRISPR, you can detect the synthetic lethality to identify the genetic dependency, or there could be, these combinations can help you to reverse the resistance. And I'm going to talk more about the reversal of resistance and how to overcome the pathway bypass to restore sensitivity. So I told you that we developed this acquired resistant model. So now we take our parental 1975 sensitive cell line and we take the 1975 acquired resistant cell line, and we perform the high throughput screening to identify the combination that have a maximum, using a high content microscopy, that can induce the maximum synergy in the cells. And we basically create this composite matrix where we can identify the drug that have highest synergy score, meaning that the drug can induce, the combination can induce maximum killing compared to both the monotherapy. So we perform this drug screen in the acquired resistant cells, and we identify that the Aurora kinase inhibitor were the top 2 synergizing agent in acquired resistant cells, along with the EGFR inhibitor. So when we validate this further using our ex vivo model, what you can see over here is that the 2 acquired resistant cell lines were completely insensitive to the EGFR inhibitor as well as 3 different Aurora kinase inhibitor. But you can see in the panel of combination, when we combine the EGFR as well as the Aurora kinase inhibitor, you see the maximum synergy in them. And when you make a xenograft of these model, and when you treat the tumor, you can see that the combination reduces the cell growth, tumor growth, in a profound manner as compared to both the monotherapy, meaning that the combination works effectively. But the question still remains, why do we see the synergy with the Aurora kinase inhibitor? What potentially Aurora kinase pathway can do? So to understand that more, I will talk about the Aurora kinase biology a little bit. So Aurora kinase consists of 3 different family, Aurora kinase A, B, and C. Aurora kinase A is mainly located in the spindle poles, and Aurora kinase B is mainly located on the chromosomal passenger complex. As you can see, Aurora kinase B in a green and Aurora kinase A on the spindle pole in a red color. Aurora kinase A is involved in regulating the survival during mitotic delay and errors. And Aurora kinase is mainly regulated by a upstream co-activator, allosteric activator called TPX2. When TPX2 binds to the Aurora kinase, it enhances its autophosphorylation and it prevents the phosphatase from binding to this Aurora kinase, meaning keeping it in a highly active form. So why we see this Aurora kinase dependency, and what does Aurora A and TPX2 does in case of a EGFR? So as you can see, we went back to our models of the sensitive models and the resistance model in red and sensitive models in green. And you can see that amongst 4 different resistant models as compared to the parental cells in green, Aurora kinase is highly phosphorylated, highly activated, and its upstream co-activator is also highly upregulated in this acquired resistant cells. So now, if we take this parental cells and overexpress them with TPX2 and phospho-Aurora, do we see increase in the cell number? And the answer is yes. What you can see over here in the panel is that without Aurora kinase A overexpression and without TPX2 overexpression, these cells are able to, only 20% of the cells are able to survive in presence of EGFR TKI. But when you give them the Aurora kinase A, TPX2, or known resistance causing gene such as KRAS is able to cause decreased sensitivity to the drug, meaning that Aurora kinase A overexpression or activation prevents these cells from drug-induced cell death. And also, when we probe this Aurora kinase activation more closely in the acquired resistant cells, it's known that Aurora kinase causes the genomic heterogeneity. And as you can see that these EGFR TKI treated cells display higher amount of genomic heterogeneity in terms of their chromosomes are highly unstable, their microtubules are completely messed up. And when you quantify them, they have like higher centrosomal defect, they have very messed up spindle geometry, and there is a lot of errors in the chromosomal segregation. But these cells are able to survive and ultimately proliferate and forms the basis of the acquired resistance. And these cells display high genomic heterogeneity. And this genomic heterogeneity is equivalent to what happens when you have the high TPX2 and Aurora kinase exogenously expressed. So what you can see over here is that when you overexpress the Aurora kinase A and TPX2, in the sensitive cell lines, you see that the number of defects that these cells harbor goes up, meaning that Aurora A and TPX2 itself can induce genomic heterogeneity. So now the answer is very clear that the Aurora kinase A and TPX2 can cause this genomic heterogeneity in the cells. They prevent the cell death. And so they are the one that give rise to the, they are the pathway that are responsible for the survival. And if we go back to our clinical equivalent model of resistance, what we see that in the case when you look at the Aurora kinase activation over a period of time, we see that Aurora kinase and TPX2 is highly upregulated from the late sensitive to residual model, and it's required in the acquired resistance model. This not only happens in case of a third generation EGFR inhibitor, but it happens in the first generation, second generation, as well as third generation EGFR inhibitor. And in terms of what it can do to prevent, the combination can be effective in terms of preventing the resistance. So here you can see that PC9, which is the sensitive cells, when you give them both the monotherapy Aurora kinase alone, there is no inhibition of cell growth. But when you give them the EGFR therapy, the cell, one in the orange and one in the green, you can see that the cells initially undergo apoptosis and there are very minimum cells left by the end of day 5. But then these cells are able to form the residual disease, and eventually by the end of day 20, they start proliferating again. But if you combine this EGFR and Aurora kinase inhibitor, one in black and one in pink, you can see that there is no emergence of the resistance, meaning that these combinations are potent to induce the higher magnitude of cell growth inhibition. And when you take the residual cells, meaning that the cells that initially survive the EGFR TKI therapy, one in green and one in blue, they can form this residual disease. But when you co-treat them with the combination with Aurora kinase inhibitor in green, in pink and in red, you see that there is a regression of this residual disease. So finally, how it can induce the what is the dependency that is created by the activation of Aurora kinase pathway in the EGFR cells? So what we noticed over here is that when you treat the cells with the YoPro, when you treat the cells with the EGFR TKI in the sensitive cells, they induce apoptosis. But in the case of acquired resistant cells, both the monotherapy in red does not induce apoptosis. But when you combine that with the MLN 8237, which is the Aurora kinase inhibitor, one in the last panel in blue as well as in the red, you see that there is a maximum increase in the apoptosis indicated by high YoPro positive cells. So these cells induce higher, combination induces the higher magnitude of apoptosis in these cells. And to study that, we went to look at the molecular activity that is going on in these cells. So these cells, as you can clearly see that in the case of parental cells as well as in the acquired resistant cells, the phospho-EGFR is not activated, meaning that there is no pathway reactivation. You can see that the Aurora kinase is inhibited only in the case of combination. So plus over here indicates the addition of Aurora kinase inhibitor. You can see that only in the case of combination in the acquired resistant cells, the BIM, which is the pro-apoptotic module of the cells, is stabilized, meaning that only the combination is able to undergo potent apoptosis in these cells. And hence, we know that EGFR as well as Aurora kinase pathway converges into the regulating the BIM stability. And how these cells do that is by creating a dependency. So when you have the treatment naive cells in case of a EGFR pathway pre-treated, you can see that the BIM is destabilized and these cells are able to survive. But when you give them the monotherapy of EGFR TKI, now BIM is able to stabilize and these cells undergo apoptosis. However, there are some residual cells that have higher Aurora kinase, and they can also destabilize BIM, and these cells are able to survive. But only when you combine the two pathways of EGFR inhibition as well as Aurora kinase inhibition, then the BIM is now stabilized and you see the apoptosis in them. So meaning that the rational design of the combination therapy is required for the complete inhibition of the cell proliferation and acquired resistance. So what I have told you so far is the strategy, how how the resistance is a prevalent problem and how it happens in the case of EGFR TKI cells. So when you take the parental cells, which are EGFR TKI sensitive, and you give them the EGFR TKI, there is a formation of drug tolerant cells. And these drug tolerant cells ultimately form the acquired resistant cells. What we saw is that in these cells, the TPX2 and AURA kinase axis is upregulated. And ultimately, these cells acquire additional alterations because they are highly genomic unstable. They're able to further select this multiple genomic driver and undergo something called alteration selections and display various genetic mechanisms that we see in the clinic, such as T790M or C797S, which is the EGFR pathway activating mutations, or the other EGFR independent genetic mechanisms such as MET amplification, HER2 amplification, FGFR amplification, or PI3 kinase mutations. This is what we usually measure in the clinic. So we think that the combination of EGFR and AURA kinase inhibitor can prevent this formation of drug-tolerant cells. It can when you target them at the residual disease, you can prevent the formation of the acquired resistant cells and ultimately the genetic resistance that we see in the clinic. So our combination strongly suggests the hypothesis that we can prevent the evolution of resistance by upfront targeting the EGFR and AURA kinase signaling. So to summarize, I have told you that the drug combinations serve as a versatile tool to address the limitations of therapeutic resistance. Rational combination design can reveal the underpinning of the bypass mechanisms. And in the case of EGFR TKI, we found that the activation of Aurora is associated with the resistance. And inactivation of Aurora kinase can serve as a synthetic lethal target to enhance the response of EGFR TKI. And the upfront combination of EGFR TKI along with the Aurora kinase inhibitor can serve as an anti-evolution approach to limit the evolution of resistance that we see. So now I want to leave you with the future direction and how we can design the combination therapy. So in the past, pathologists would look at the cancer samples and design what would be the drug target for the patient. In case of the modern genomic era, we rely highly on the protein expression, mRNA expression, or single gene amplification, such as what is the genomic driver. But in future, we will be able to do the deep profiling and identify what are the different genomic drivers or proteomic drivers that are present that drives this, and that can inform the rational design of combination therapy. Currently, we perform the drug combination design was just based on the hypothesis or just based on the model of calculating the combination index. But with the advent of precision oncology and with the decreasing cost of NGS methods, we can generate this knowledge-based evolutionary system-based model to identify what are the pathway regulations and what it means in terms of therapy. Initially, we relied just on the chemotherapy, but now with the advent of present technology, we know what are the genomic drivers, and we can combine that knowledge to design the efficacious combination. In the past, the drug resistance was only based on the efflux transporters that are present on the cell surface, which will limit the efficacy of the drug. But now we know how to classify the patients according to their genomic underpinning, and hopefully that can help us to design the better combination in terms of if there is a resistance mechanisms that are present, and hopefully that would be able to design adaptive clinical trials that will classify patient based on their genomic driver of the resistance and allow the patient to undergo the combination therapy. So that's a vision for the future. But there are limitations to this combination therapy, meaning that there are various challenges in terms of validating the combination. The target validity and the target engagement need to be validated. The pharmacological effects of combining both the therapeutic modality needs to be validated. How do we select the patient who will derive the maximum benefit from the combination is unknown. There is sometimes pathway overlapping toxicity that will limit the use of the combination. We need to design the optimal dose and the dosing schedule. We need to identify biomarker of response of the patient who will respond to this combination therapy, or what would be the resistance mechanisms to the combination therapy is need to be investigated. So that's the future directions, how we can utilize the combination in drug discovery. So with that, I would like to say thank you all. I would like to thank my lab members at UCSF and various collaborators who makes this as such an exciting work environment, and my funding agencies. Thank you all. Part of this work was published in Nature Medicine, so please cite our work and let me know if you have any questions. Thank you so much.
Thank you, Dr. Shah, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May of 2019. And as a final reminder, our speaker will follow up with any of your questions you've submitted via email. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.